Hi, everyone. Welcome to Foresight's Biotech and Health Extension Group, sponsored by 100 Plus Capital. I'm really excited to have Jean-Hebert back with us again. I think it's maybe your third appearance here in one of the seminars, and we've definitely had the luck to have you on for a lot of the workshops. I think we got in touch for the first time a few years ago when you wrote a really amazing book on the replacing paradigm to aging. And yeah, you've since done a lot of research and now also have a really wonderful company underway. And so we thought that it would be really fun to invite you back, given that replacing is now really mentioned, I think, in the Amaranth Foundation and their longevity bottleneck assessment in the longevity biotech fellowship roadmap. And so I think it's just popping up a lot as a theme that is pretty undervalued within longevity, but like really promising. And so I'm really excited to have you here as, to me, the pioneer who really like first set the whole entire kind of like subdomain within aging on the map, at least for me. So thanks a lot for joining, John. You'll be discussing replacement as the surest, fastest, and cheapest way to beating aging. And I will share a lot more about your bio in the chat. But without further ado, thanks a lot for joining, and the stage is yours.
Thanks so much, Allison, for having me again. Hope you can hear me well enough. Yeah, no, it's a pleasure to be back. And it's nice to see that replacement is growing in appreciation in the field. I think it's really because it deserves more attention, and hope maybe I'll convince you of that today. The title is a bit provocative, but I think it's totally defensible. And happy to discuss this. I'll show a few slides and open to this being interactive and stimulating discussion about this. I'm going to be telling you about replacement and why I think it's the surest and fastest and also cheapest way of beating aging. So first of all, why even consider a replacement as an approach to life extension? Let's start with an example. I got this from Anar Isnan, who posted this recently, but I think it's a pretty illustrative picture. This is the same person decades apart. Some of you may recognize this person, Clint Eastwood, well-known actor and director. But this is what aging does. And what exactly is the difference that we're looking at here between the same person decades apart? And really what it is this accumulation of damage that occurs. It's stochastic, it's complex damage. I've listed some of the forms of damage that accumulate to protein over time here, especially to long-lived proteins. These are just classes of damage, so they each break down into different specific types of damage to the proteins. So it's very complex. It happens somewhat randomly. This is not, programmed. And most importantly, it's non-enzymatic, meaning it's not a biological process. It's really a physical process of the breakdown of long-lived proteins over time. And this is particularly important because approaches like drugs that are targeting biological pathways, aren't going to have much of an impact on this physical process. Also, we pretty much know all the genes in the genome now, and we can say with confidence that there are no genes encoded in our genome that recognize this damage or that encode the machinery that can deal with this damage. So genetic approaches and epigenetic approaches are not really going to have much of an impact. You might say, oh, there are, for these extracellular long-lived proteins that accumulate this damage, there are things like collagenase. It's known that collagenases stop working on these proteins when they accumulate damage. So we're really left with nothing in the cell that can deal with this. And we know that this damage that accumulates around and outside of cells really has a big impact on the behavior of these cells in terms of whether they're behaving like young cells or old cells. So these are called heterochronic transplants, where you can take cells from a young tissue and then transplant them in an old tissue, or vice versa. And what matters is, the end tissue that the cells end up in. So young cells in an old tissue behave like old cells. old cells in the young tissue behave like young cells. We can kill ourselves trying to make these cells young again, which I would argue most of the longevity field is still focused on. And that's really not going to have much of an impact unless we deal with all the extracellular components as well. And the only way to do that at this point, or at least the most direct way, is to do tissue level or greater replacements, organ level, tissue level, whole replacements. Replacements are already used in the clinic, so there's not too much of a hurdle there. They're used to reverse damage caused by trauma, for example, or to cure certain diseases. But of course, they could be used for a lot more if we're thinking about life extension. And so I'll go over, the different types of replacement approaches, both for the body and the brain, and the pros and cons, and then this could potentially stimulate discussions about, some of I probably missed some of the pros and cons, or definitely open to feedback on that. But first, I just wanted to make a sort of a side-by-side comparison, again, with much of the focus of the longevity field versus replacement. And so what are the advantages of taking a replacement approach? A lot of us have heard that, we have to understand aging before we can address it. Maybe, but not if you're using replacement, where you do, wholesale tissue replacement. All the damage is reversed at once, and you don't really have to understand, the thousands of things that are going wrong with aging to be able to reverse that. Aging biomarkers, a similar idea. You can find limitless numbers of things that change with aging and call them biomarkers and say, they're more or less important. But again, you don't really need that when you're doing tissue or greater level replacements. Another aspect of or focus of the longevity field is to really get the FDA to approve drugs as anti-aging drugs, to get them on board with longevity research. And, that's great. The more people we get on board with longevity research, the more we get government regulatory entities on board with the idea of life extension, the better. But for replacement, we don't actually need that. Replacements are generally approved. We've seen recently in the news, for example, a woman just got, 2 pig organs transplanted at the same time, kidney and thymus. And previously, the other individuals have gotten also xenotransplants for other organs. And of course, tissue engineered organs have been used in the clinic. And of course, a lot of donor to recipient organ transplants, and every body part has been transplanted that way in people. So the FDA is very much on board with these replacement strategies already, even though right now they're for disease and trauma, but in general, there's no restrictions there. Also, getting more scientists interested, again, that's very important. Nothing wrong with that. But in terms of replacement and this regenerative medicine approach, there's a ton of talent out there. It's one of the hottest areas of research in biology. And right now, there is certainly sufficient talent there. It's just a matter of funding to hire them. The costs of, going about extending lifespan by targeting all the different pathways involved and trying to modify them are indefinite at this point. This is largely based on work done by the LDF, the recent LDF roadmap, where they went out and talked to a lot of people about, how much it would cost to get to particular milestones and how long it would take. And the cost is indeterminate because the timeline is indeterminate, and there would be lots that would need to be addressed. In the case of replacement, the milestones are very well defined for the different approaches, which we'll go over again in a minute. And so you can come up with cost estimates for that, which are, significantly less. again, thank you to Leah, Mark Hamalainen in particular for putting that together. Yeah, and then of course, we don't even know with all these efforts, and I would argue that the probability of success with these efforts is very low, whereas with replacement, we know if we succeed in doing it that, if you have an old body and replace it with a young body, you will have reversed aging. It's very high probability of success. Okay, so I don't know if there's any questions yet at this point. Happy to answer.
No, you're good.
Good. All right. So let's start with replacement for the body. And there are several possible options of how we can go about it. In this case, there's biological, two broad categories, biological, non-biological. And then within the biological, you can do full body replacement. You can do all internal organs at once, or you can do more, part by part, organ by organ replacement as well. So those are different approaches, and we'll touch upon each of those. Then a non-biological, similarly, you could do potentially, full body or all organs, or part by part as well. And, there's efforts going on for all these approaches and, initial early proofs of concept that it can be done. So whole body replacement, it's conceptually the most straightforward. I think it's also technically the most straightforward because it really doesn't involve any new tech that doesn't exist. The tech has to be adapted to this. But again, the proof of concepts are there that the technology could work for this. But it would involve taking these primitive cells, germ cells, or other primitive stem cells, and just growing brainless bodies. Obviously, we wouldn't want, this to be unethical in any way. And so if there is no brain there and it's just a body, it should be okay to use. And then, you would use that to replace an old person's body with a young body. So basically you would do a body transplant. So the old head would be on a young body. The pros for this are everything except the brain is young at once, in one shot. There's potential for immune matching depending on where these cells come from that you're using to make the body. And as I mentioned, it requires little new tech. Proof of concept in preclinical models has been done for all these steps. The downsides are, the social resistance. People, hear this and go, "Oh my God, this is crazy." And so I think that's a hurdle that, needs to be addressed. And you also need to deal with the severed spinal cord when you do a head or brain transplant. Although there too, the technology is advanced quite a bit to either bypass the spinal cord severing, for example, using brain-machine interfaces, which are in clinical trials, or biologically regrowing those connections, where a lot of progress has been made as well, although, cannot claim success on that yet. And also scaling is a consideration here that is not necessarily the easiest to address. So another approach that is being considered and explored is simply replacing all internal organs at once. It's actually not that complicated a surgery because there are not too many tubes at the top and the bottom of all these organs. So the surgery would appear to be doable, although that has not been tested yet. But it is an approach. The advantages in this case, instead of doing like the whole body transplant, is that there's no spinal cord severing. And there's still possibility for immune matching, and also requires little new tech. But here too, the social acceptance of such an approach might require a fair amount of, convincing that this is going to be saving lives and no one's getting hurt and, that this is really the right thing to do. But that is definitely, I think, something that would still need to be addressed. In this case, not all body parts are replaced. It's just the internal organs, which you could argue would likely have benefits on health. But, and I put this in the cons because it's not all body parts. And I would worry, though, because, even like the limbs, for example, that are aging over time put you at risk of death. As their blood vessels, get old, you're more likely to get clots, which can result in pulmonary embolisms and kill you, for example. Maybe it would extend a maximal lifespan, which would be great. Not necessarily. You still have to consider those other body parts. Scaling, again, here too, is not something that is obvious, although, there are solutions. So the other approach is to do more organ by organ or part by part. And we've seen a lot of evidence of this in a clinic already, as I mentioned. Recently, xenotransplant, where you basically use normal developmental processes to generate organs of the right size, which is why pigs are used. And then if you can deal with the immune incompatibility, then you can use these for transplantation. So that's an approach that's getting a lot of attention now and is being used in certain cases in clinical trials with certain amount of success. Those organs are working in humans, but still a ways to go there in terms of reducing the immune rejection risk, among other things. And in this particular case with xenotransplantation, one of the downsides is, you're still using animals for this. Although again, there may be a way around it because, in preclinical studies, we do know how to make non-sentient bodies. And so that could be a way of resolving that issue. In any case, there's also other approaches like blastocyst complementation. So in this case, you would be growing human organs in another species like the pig, again, for size compatibility. But in this case, the organs would be made of human cells. And then, of course, there's what, the field has been working on for quite some time with fairly slow progress is tissue engineering. And that's where you basically make de novo organs in the lab that can be used for transplantation. And there's been some success, like the bladder that's already probably a couple of decades ago, or at least a decade and a half. And, those are functioning in humans quite well. And so that's a success. But those have been far and few in between. And I think the advantage of these other approaches, like the xenotransplantation or growing human organs in a pig, for example, is that they don't require really, other than genetic engineering for the immune compatibility, they don't require tissue engineering because it's really letting nature or the developmental process occur on its own as it normally does. And I think that's a concept that is really going to take over the replacement field over time. It's just using normal developmental processes to generate the tissues, the organs that we want for replacement. Because we don't, again, have to understand all the steps or, we don't have to be able to de novo make things from scratch. They will just grow and develop naturally as they normally would. When we were fetuses growing up and becoming kids and growing up into adults. Yeah, in this case, the advantages, again, no spinal cord severing. Scaling could be easier. Again, depends on, which of these approaches you're taking. And in all cases, it's already happening. So there's early proof of concept that this can work. The downsides, if you're going to do it this way, it means a lot of surgeries, which, again, puts you at greater risk of complications. And it's hard to replace all body parts this way, although conceivably you could. And the immune mismatching, again, that can be different depending on the approaches here, but it is a challenge with using these different parts. All right. So there's also a non-biological approach to replacement where you can do replace like all the organs of the body with synthetic replacements. And I only know one lab who's doing this, but they seem to be doing quite well. This is, the Brain X from Stan's lab at Yale. And so they have this completely synthetic artificial system that replaces the need for all our organs. And, of course, because this is engineered, it could also easily be replaced when it gets old, or the parts can be maintained, much more like an old car where you can keep it going indefinitely if you change all the parts. So this is an interesting approach that, I'm keeping an eye on. We should all keep an eye on. The advantages are easy scaling, and there's opportunities here for brain-machine interfaces to combine that with controlling limbs, for example. I think that's an advantage. I also put it in the disadvantages because brain-machine interfaces are required in this case to, be able to interact with the outside world. And so the state of the art for these brain-machine interfaces are getting pretty good for the machine reading your intentions and being able to control limbs, whether they're artificial or your own limbs, if you're paralyzed, for example. So they're getting pretty good at that, or they are pretty good at that already. But there's very little in the way of sensory input to the brain using machine interfaces. And so that's an area, I think, where before this can be useful, that would need to be addressed. And then, potentially you still have to worry about the other body parts that are biological that you haven't replaced. But, that could potentially be addressed, again, using synthetic body parts like synthetic limbs, for example, which have become very sophisticated and perform, almost as well, or maybe in some cases better than our natural limbs, certainly better than our natural limbs as we get old. So that's also an approach that I think is of interest. And of course, there's a fully artificial heart that's being used now in people. There's also kidney like bioreactors that are in preclinical stage but are working pretty well. And other organs potentially could also follow suit. So you can do this replacement with synthetics, either part by part or the whole thing. And, the brain remains biological. Maybe other parts of the body would be biological as well if you were doing this in combination with biological replacements. So there's a bio-machine, the compatibility is not always perfect. And so there's a risk there. I think the technology can stand to improve a little bit. It's gotten pretty good with blood vessels, which are really important because, if the biological vessel detaches from the synthetic one, it can be pretty disastrous. But I think a lot of progress has been made there. But, overall, I think still some progress to be made. So that's the body replacement approaches. I'm going to move to the brain, but unless anybody has questions in the meantime. There's
one, but I think we can move on and then we'll tackle them all one by one.
Okay, great. So brain tissue replacement. Obviously, we can't replace the whole brain at once. It would be a different person. So we have to consider how we do this. And the only way would be progressive tissue replacement. And again, in terms of FDA approval in the clinic, this doesn't seem to be a problem using cells, or I wouldn't call them tissues yet, but they're combinations of, scaffolds and cells that are being used. So you call them very primitive tissues that are used in people already. So, there's no roadblock there in terms of getting it to the clinic. Here's some successes in terms of treating Parkinson's and epilepsy with new cells. These companies like Aspen, Neuroscience, Blue Rock, Neurona. Aspen, not yet. They have FDA approval, but everybody anticipates that their cells are going to be as good as Blue Rock's without the need for immune suppression because they're autologous. But these companies have shown safety in the clinic and efficacy of their cells in these brain transplants. And, they're also commercially very viable successes. So the companies are worth a lot at this point. Again, because it works. Their cells work to reverse, these, in the case of Parkinson's, age-related disease. And in the case of epilepsy, just a neurological disease. That's just to say that, that there's a path to getting this implemented in the clinic. But there's two reasons why progressive brain tissue replacement makes sense. One is that it appears that we'll be able to add new tissue to the brains and have it functionally integrate with the existing brain. This was shown most nicely, already 8 years ago in Germany by Magdalena Goetz's lab, showing that transplanted embryonic precursors differentiate into neurons that integrate remarkably well with the adult neural circuitry. So very promising result there. And then this was repeated and continues to be repeated by a bunch of different labs, including ours, just showing that, you can get really good integration of new neurons in the brain. Whether you're transplanting dissociated cells or organoids, they all seem to do quite well. Yeah, I just have some examples here, and they all basically say the same thing, that the neurons integrate really well. However, we're not there yet in terms of those neurons truly being functional, because there's things missing. So these are typically a single neuronal type, or at least a single neuronal a single class of neurons. For example, excitatory neurons and not inhibitory neurons. And so we know that without the full complement of neuronal subtypes, that tissue or that graft cannot process information normally. It also lacks structure. So we know the structure is really important for the wiring within the tissue, or in this case, within the graft. And so if the cells are all mixed up, which they are in all of these publications, including in the work we've done and published, you're not going to get information processing that's useful to the host. We still have work to do. But again, this is, doesn't require development of new technologies. It just requires doing things right. But so this shows, at least in principle, that we should be able to add new tissue to the old brain and have it functionally integrate. But important to progressive tissue replacement is not just the addition of new tissue, but the removal of old tissue, which inevitably, lead to strokes and other bad things. So we need to get rid of that tissue. And we already have a proof of concept in the clinic that old tissue can be removed without loss of its information content or interruption of function or personality or self-identity in cases slow-growing benign gliomas that occur in people of any age, but typically more so as you get older. So even in patients in their 70s, for example, they get these tumors that grow from a pinpoint out and eat away a certain area of the brain, or the neocortex in particular. And let's say it eats away at the language center over the course of an extended period of time. Those individuals don't lose the ability to speak, because language gets re-encoded in a different part of the neocortex because of the progressive nature of the destruction of the tissue, and because the person is using language every day. If you have a stroke in the language center, it's a catastrophic event. There's no time for the relocation or the plasticity to occur, and you lose language. But these tumors show that you can, lose tissue slowly over time without losing the information content in it, without you even realizing that it's happening. So this is the other half of the equation. We can add tissue, and we can progressively remove tissue without loss of information content. So great. How might we go about doing this? There are different here too, just like for the body, for the brain, there are different approaches. There's an engineering approach where, again, you're in all cases inspired by the normal developmental process. So you want to recapitulate that normal developmental process. And this is something that my group is working on, engineering a fetal-like neocortical tissue, which is very well defined after doing extensive omics analyses to know what precursor cell types are present there, what are the extracellular signals and scaffolding components that are necessary for those cells to develop into a well-structured, mature tissue that can process information normally. So we've done this analysis. We can generate all the components. Sorry, this is a bit of a plug for the work we're doing, but it's only a couple of slides. And so we've actually generated a proto-tissue now, has the structure and the precursor cell types that we want. And we've started testing this in animals. Again, still a lot of work to do, but we think it's possible to engineer this fetal-like tissue to give rise to structured, functional adult tissue. It's not the only approach, though, although it does have advantages in terms of surgical implementation, because since we're designing the tissue, we can design the shape and adjust it to each lesion site, for example, and it's much easier to scale than the approaches I'm going to tell you about now. The disadvantage is it's harder to get it right because compared to the advantages I'm going to show you now as well. And those approaches are basically to use normal development to obtain the same precursor tissue, in this case, not engineered, but derived from earlier developmental stages. So you may have heard of these ex utero synthetic fetuses that different groups are working on. So they start with human pluripotent stem cells in culture, which, standard practice now in terms of deriving those from individuals, and then developing them into these sort of early fetuses. And the big advantage here is that those tissues are more likely to be authentic. This is also scalable, which is nice. The disadvantage is the need potentially for synthetic uteri to get to older stages where we could actually collect this fetal-like tissue for our grafts instead of engineering it, for example. And that hasn't been developed yet. Otherwise, it may be hard to get these synthetic fetuses to develop to a stage that's old enough that we could collect tissue without necrosis setting in. People are working on it, and they're smart people, and hopefully they'll succeed soon. Another way is to use interspecies chimeras. There were 2 papers that just came out this month showing that you could grow, I think it was rat cells in mice and get, in both cases, it's not complete, so it's not clean. I think there's ways that you could make this a lot cleaner. But in red here is a rat brain tissue in an otherwise mouse brain. And in another study, they show a similar thing, although the degree of chimerism, in a way, is worse. I think this could be improved, and you could imagine growing human fetal-like or human fetal tissue, brain tissue, in another species to obtain a source for transplantation and repair. And then you could use human fetal tissue itself. This has been used for quite some time, for Parkinson's, for example, in 1987, 88, and then ever since, human fetal brain tissue has been used to treat Parkinson's in particular. And, one could imagine that using human fetal tissue could be a source of tissue for transplantation. Disadvantage, there is some social opposition to using human fetal tissue, even though at this point, there are not even any functional neurons there. So it's really not, there's no sentience. And the scaling is also, problematic. But in any case, that's an alternative approach where we know the tissue is truly authentic. So I think that's pretty much it. For the brain, this is just a cartoon showing how, the addition and removal of old tissue could be implemented. We have ways, although they haven't been adapted to our purposes yet, but we have ways of progressively silencing brain tissue from a pinpoint out that would mimic a glioma, because we wouldn't want to use glioma to silence tissue, as an example I showed you earlier. But we have ways of doing that. So you could progressively silence parts of the brain while putting in your fetal tissue in other areas and allowing progressive silencing and tissue development and integration occur coincidentally, and repeating this a few times to turn basically an old brain into a young brain. I went over that fast, happy to go over it in more detail or where it's really interesting. But the point is, none of this requires new technologies. It's all adaptation of existing technologies. I think it's all achievable, and combined with the body part replacements or body replacements, we could beat aging in a reasonable amount of time and with a reasonable budget that isn't crazy. So anyway, open to feedback and questions at this point.
Fantastic. Thank you so very much. That was really interesting. And I love that. I think in your previous talk that you gave at Foresight was mostly really focused on the brain. And now I think, going through the whole path of the whole body first, I think laid a really great vision out for that. There's a ton of questions already, so I'm going to keep mine very brief, aka I'm going to ask them at the end in case we have time. But for now, we have Fiona. Why don't you kick us off?
All right. Thank you, Alison. Jean, thank you so much. This is fascinating as always. My question is about the brain replacement, right? Somewhere I read that a brain that doesn't evolve or grow up with the body, where it maps how to control, let's say, fingers, limbs, and all that, if it's placed in another body, it will not know on the lower levels of the brain function what to do, and that the organism will die. Also, I understand there are neurons outside the brain, in the heart and in the stomach. And, do these have memories that build up over a lifetime? Can you address that?
Yeah, I don't. There's certainly no conscious memories there. I like to use quadriplegics as an example. They don't, after their accident, for example, they don't lose certain memories that would be in the neurons that are in the rest of the body, for example, right? So they're still the same person. They may be a little bit depressed for a while about their new condition of being paralyzed, but essentially it's the same person, and they're not connected to any of the neurons in the rest of their body. So in their digestive system, their sensory neurons, motor neurons, all the different types of neurons that occur in the body. So I don't think those neurons really contribute to who we are in terms of our personality, or at least not directly who we are in terms of our personality or self-identity. Yeah, the other question was, the connection again. Yeah, so it's the same thing. You can be disconnected neuronally from your body and your brain doesn't die. And during development, the process is coincident with body growth and those neurons connecting to the body. But, my understanding of the physiology of the neocortex is that it is superplastic. And you can, for example, if you get a new limb, that you can somehow communicate with your brain, you incorporate that new limb as yours. So there's so much plasticity in the neocortex, it won't know the difference whether, if you get a synthetic limb, because you lost a limb and you get a synthetic limb, if there's some proprioceptive feedback from that artificial limb, your brain, your cortex, will adapt it as if it's your limb. So I think there's enough plasticity there to, connect with the world normally.
All right. Thanks, Aaron.
Hey, this is nitpicky, but I feel like it's justified. So the slide that you had for your arguments about why the replacement should be done over other things. For understanding aging and for biomarkers, if we don't know how maintenance occurs for these replaced portions, at least at the tissue level, then your same argument about a cell being in a bad environment leads it to being an old cell still applies to tissues. So if we don't understand how maintenance occurs, then tissue level replacement, it might be that you replace the tissue and it fails within months because the maintenance systems were turned off. The logic that you outlined is all correct for whole body replacement if you grow like a whole entirely new body. But for tissue level replacement, which is the title that you used on that slide, I don't think it's correct. So if you can update that portion, that'd be good.
Yeah, I did I say just tissue level or tissue or greater level? I can't remember. Sometimes I use both, but yeah. So I'm trying to be all inclusive about the different approaches. But yeah, it's a good point, right? That if you have, and certainly if, if you have a single organ, young organ transplanted in an old person, it's unlikely to do much good, right? Because failure of any other organ is going to kill not only that organ, but the whole person. The effect of the surrounding environment, though, to cells, I think it's something, yeah, you could explore. Biomarkers could be useful for that, for sure. The evidence, even in the brain in humans over time, is actually pretty good that the environment doesn't, hurt the new cells in that case that much. And if we're talking about tissue level replacements, it should even be a lot less because the cells in the new tissue will be much more buffered from the rest of the environment because they're surrounded by the young tissue. But in the cases that I'm referring to, again, from back in the 80s, when they were using fetal human brain tissue to treat Parkinson's, they did post-mortem analyses of these, up to 2 decades later. And even though those were naked cells in a diseased environment, accelerated degenerative environment, after 16 years, they still looked pretty good and were still functioning. And then after, over 2 decades, they were starting to show signs of pathology. And again, those are naked cells in a diseased environment. And they still lasted, close to a couple of decades. I think we can be relatively confident that the environment does matter, of course, like we mentioned, because you do need to replace everything eventually, and probably in a reasonable amount of time. But I think, I think there's pretty good evidence, and yes, it could be confirmed with biomarkers that young tissue is doing pretty well in an old environment. We have a collaboration with Radin's lab where we started to look at this. And yeah, obviously he works on biomarkers, epigenetic biomarkers. And so I think it can still be useful. I would still argue that I don't think they're necessary though, but yeah.
Next one we have John.
Hi, Jean. Nice presentation. I particularly like the way you outlined the importance of extracellular aging as contrasted with cellular aging. I'd like to suggest a couple of alternative approaches that might run in parallel with your whole body or whatever replacement. One is to genetically engineer fibroblasts and fibroblast-like cells so that they would go through more quickly and replace aging collagen, aging elastin, and so forth. Right now they do it, but they take around 7 or 8 years in most tissues to replace the collagen. And if we could speed that up, that could be very therapeutic. The other is drug alternatives to breaking crosslinks, glycation crosslinks. There's one drug that was developed 25 years ago, allogebrium or ALT-711, and that works on one kind of principal crosslink, the alpha diketone. There's another principal crosslink, glucose pain, which so far we don't have a small molecule that can attack that. So I wanted to suggest that we expand your project to include some parallel projects.
Yeah, I'll start with the second question first. Yeah, that is a molecular approach. I wouldn't say it's impossible. I just think it's very difficult because of the complexity of the damage. There may be a couple of examples of drugs that can reverse crosslinks, but I'd be very surprised if they had no side effects, because it's very hard for a drug to distinguish, a very similar covalent bond being a bad protein or versus a good protein.
In the case of alpha diketone, they don't exist inside the cell, and they don't have any particular beneficial purpose. So breaking the alpha diketone links could be done without side effects. I've been taking allogebrium for 24 years now, so I'm pretty sure it's pretty safe. From your initial slide, of course, you showed that another problem is just breaking of elastin and breaking into the strands. And that couldn't be fixed by allogebrium, but it might be fixed if we can genetically engineer a new kind of fibroblast that will chew up the old extracellular matrix and lay down new extracellular matrix in its wake.
Yeah. And to your first point, I think, that makes sense if it's something that doesn't exist normally, for sure. But there are so many other, modifications that occur over time. Breaks, like you mentioned, but not only breaks, there's aggregates, there's lipid cross-linking, there's carbohydrate cross-linking, protein cross-linking, or different types. There's calcification, there's, it's, maybe you can imagine drugs for all of these, but again, because of the stochastic nature, I think it would be very difficult.
I agree 100%. I do think that it's important to work on this parallel path along with your project.
Sure. The other question with the fibroblasts, engineered fibroblasts, some collagens turn over collagens are a big family, right? They're the most abundant proteins, if you put them all together in our bodies. But there are a lot of different types of collagen. Some of them will turn over relatively fast, some very slowly, and some almost not at all. But in the cases studied, whenever they start accumulating damage, they're no longer turned over by collagenases. So I'm not sure how putting in new fibroblasts is going to help that. They have the same collagenase as the old fibroblasts.
I believe that the fibroblasts will actually chew up the old collagen, not just secrete collagenase. That's my understanding.
Yeah, I don't know how phagocytic they are. Typically, it's more, immune-derived cells that do the bulk of that, even when there's fibrosis. They certainly sound the alarm. Fibroblasts will become senescent when it's damaged. But yeah, there might be something there. I'm not familiar with that area of fibroblast, PCM.
We only have, sorry, John. Yeah, please share. Yeah, please share a link, John. We only have 1 minute, and I really want to make sure that we also get your final words on what it is that you're doing right now, Jean, and how people can help and support you if they're interested in doing so. Because you showed a few slides about the company, but it would be useful to have some kind of clear action items for people to take away with. That would be great.
Yeah. Sorry, Micah, I saw you had your hand up. I would have loved to hear your question because you always have good questions. But maybe you can, hit me later. Yeah, I think just being here, discussing this more is a positive thing. Happy to see that. We're looking for people who want to get involved with these replacement approaches. There's a lot of work to be done with progressive tissue replacement in the brain. So anybody interested in that, please reach out to me, see if we can work together. That would be great. But yeah, that's it. Thanks for having me. Yeah.
All right, great. I'm hoping that we hear a lot more as you guys are gradually building out things. And please do get in touch with Jean. I think his work is absolutely fantastic. You've made so much progress in such a short time. I think there's so much to do as you laid out in the different categories. But I really think that, you're moving really fast, and it would be great if more people can jumped on and supported this type of work. Thank you so much. Thanks everyone for your amazing questions. I shared your Twitter here that people can contact you through, but you also shared your email address, I think, in the final slide. or in the first slide so that people definitely know how to contact you. If you have trouble contacting Jean, contact me to contact Jean. And yeah, I'm really excited for what you guys are building. And I think there could hardly be enough support, I think, for this type of stuff right now. We need to move fast. Thanks a lot.